Bismillah ar-Rahman ar-Rahim. Assalamu alaikum wa rahmatullahi wa barakatuh. Anatomy and Physiology, 2026. Chapter 15, The Special Senses. Lecture outline. The special senses include olfaction, taste, vision, hearing, and equilibrium. 15.1 Olfaction. Olfaction, sense of smell. Olfactory epithelium found in superior nasal cavity in the olfactory region. 10 million olfactory neurons. Dendrites of olfactory neurons have enlarged ends called olfactory vesicles. Olfactory hairs, olfactory neuron cilia. Odorants dissolve in mucus attached to receptors. Cilia depolarize and initiate action potentials in olfactory neuron. Receptor may respond to more than one odorant. Olfactory region, epithelium, and bulb. Olfactory bulb, olfactory tract, cribriform plate of ethmoid bone, cut edge of epithelium, nasopharynx, frontal bone, fibers of olfactory nerve, olfactory region, nasal cavity, hard palate. Medial view. Posterior olfactory tract, cribriform plate, connective tissue, olfactory epithelium, mucus layer on epithelial surface. Secondary neurons of olfactory bulb, foramen. Olfactory nerve processes, axon, basal cell, supporting cell, olfactory neuron, dendrite, cilia, olfactory hairs, olfactory vesicle. Posterior, anterior, medial view. Action of odorant binding to membrane receptor of olfactory hair. Odorant receptor. 1. Odorant receptor. G protein. Odorant. Adenylate cyclase, Na+, gated ion channel closed. 2. Odorant binds receptor. 3. G protein activates adenylate cyclase. 4. CAMP produced and binds to channel ATP CAMP. 5. Ion channels open. Action of odorant binding to membrane receptor of olfactory hair. 1. Each odorant receptor molecule is associated with a G protein. 2. Binding of an odorant to the receptor molecule activates the G protein. 3. The G protein activates adenylate cyclase. 4. Adenylate cyclase is an enzyme that catalyzes the formation of cyclic AMP CAMP from ATP. 5. CAMP in these cells causes Na+ and Ca2+ channels to open. The influx of ions into the olfactory hairs results in depolarization and the production of action potentials in the olfactory neurons. 15.1 Olfaction. Olfactory receptors. Receptor molecules vary in structure to allow for about 1,000 different odorant receptor molecules that react to odorants of different sizes, shapes, and functional groups. Use multiple intracellular pathways involving G proteins, adenylate, cyclase, and ion channels. Average person can detect about 4,000 different smells. Threshold for detecting odors is very low, and adaptation occurs quickly, replaced about every 2 months from basal cells in the olfactory epithelium. 15.1 Olfaction. Primary classes of smells: camphorous, mothballs, musky, floral, pepperminty, ethereal, fresh pears, pungent, putrid, maybe as many as 50 primary odors. 15.1 Olfaction. Neural pathways for olfaction: olfactory sensory pathway, olfactory neurons bipolar in the olfactory epithelium pass through cribriform plate to olfactory bulbs and synapse with tufted cells or mitral cells. These extend to the olfactory tract and synapse with association neurons. Association neurons also receive input from brain, so information can be modified before it reaches the brain. Information goes to olfactory cortex of the frontal lobe without going through thalamus. Only major sense that does not go through thalamus. Olfactory cortex and neuronal pathways. 1. Axons from the olfactory neurons, which form the olfactory nerves, cranial nerve. 1. Project through numerous small foramina in the bony cribriform plate to the olfactory bulb. 2. Within the olfactory bulb, the olfactory neurons synapse with secondary neurons which relay olfactory information to the brain through the olfactory tracts. Olfactory bulb neurons also receive input from nerve cell processes entering the olfactory bulb from the brain, enhancing adaptation that occurs along this part of the olfactory pathway. Olfactory cortex and neuronal pathways. Frontal bone, olfactory bulb, fibers of olfactory nerve, nasal bone, nasal cavity, olfactory tract, olfactory cortex areas of the cerebrum. 1. Olfactory neurons extend to olfactory bulb. 2. Olfactory bulb axons extend to olfactory cortex areas. Medial view, figure 15.3. 15.1 Olfaction. Olfactory processing. Majority of neurons in the olfactory tracts project the central olfactory cortex areas in the temporal and frontal lobes, where they are processed to allow us to perceive odors. Includes the piriform cortex, amygdala, and orbitofrontal cortex. Secondary olfactory areas involved with emotional and autonomic responses to smell. Includes the hypothalamus, hippocampus, and limbic system. 15.2 Taste. Taste. Gustatory stimuli detected by taste buds, located mostly along edges of papillae. Types of papillae: filiform, filament-shaped, most numerous, no taste buds. Velate, largest, least numerous, 8 to 12, in V-shape, along border between interior and posterior parts of the tongue, have taste buds. Foliate, leaf-shaped, and folds on the sides of the tongue, contain most sensitive taste buds, decrease in number with age. Fungiform, mushroom-shaped, scattered irregularly. Fungiform, mushroom-shaped, scattered irregularly over the superior surface of tongue. Have taste buds. Papillae. Figure 15.4. Posterior epiglottis, palatine tonsil, epithelium, taste bud, C. velate papilla. Anterior, posterior, anterior. B. filiform papilla, surface of the tongue, epithelium, taste bud, D. folate papilla, E. fungiform papilla, epithelium, taste bud, E. fungiform papilla. Nerve fiber of sensory neuron. Basal cell, supporting cell, taste hair, taste pore, epithelium, F. taste bud. Synaptic vesicles, nucleus, taste hair, sensory neuron terminal, G. taste cell. 15.2. Taste buds consistent of three major cell types. Taste buds consistent of three major cells types: taste cells, basal cells, supporting cells. A taste bud has about 50 taste cells, each having several microvilli called taste hair. Taste hairs extend from apex of the taste cell through a taste pore. Taste cells are replaced every 10 days. Nerve fiber of sensory neuron. Basal cell, supporting cell, taste hair, taste pore, epithelium, F. taste bud. Synaptic vesicles, nucleus, taste hair, sensory neuron terminal, G. taste cell. Figure 15.4 FG. 15.2 Taste. Function of taste. Tastants dissolve in saliva, enter the taste pores, then stimulate the taste cells. Texture and temperature affect the perception of taste. Very rapid adaptation at level of taste bud and within CNS. Taste influenced by olfaction. Different tastes have different thresholds, with bitter being the taste to which we are most sensitive. Many alkaloids, bitter, are poisonous. All taste buds can detect all 5 tastes, but are usually more sensitive to one. 15.2 Taste. Classes of tastants. Sour, most sensitive receptors on lateral aspects of tongue. Salty, most sensitive receptors on tip of tongue. Anything with Na+ causes depolarization plus other metal ions. Bitter, most sensitive receptors on posterior aspect. Highest sensitivity. Sensation produced by alkaloids, which are toxic. Sweet, most sensitive receptors on tip of tongue. Sugars, some carbohydrates, and some proteins. Umami, scattered sensitivity caused by amino acids. 15.2 Taste. Stimulation of taste receptors. Salt, sodium ions diffuse through Na+ channels, resulting in depolarization. Sweet, sugars such as glucose or artificial sweeteners bind to receptors and cause the cell to depolarize by means of a G protein mechanism. The subunit of the G protein activates adenylate cyclase, which produces CAMP. CAMP activates a kinase that phosphorylates K plus channels. The K plus channels close, resulting depolarization. Stimulation of taste receptors. A salt and A plus channel protein. B sweet, sugar or sweetener. G protein coupled receptor. Adenylate cyclase. K plus channel. ATP CAMP kinase. P. Figure 15.5. 15.2 Taste. Stimulation of taste receptors continued. Sour hydrogen ions of acids cause depolarization of taste cells by one of three mechanisms. Enter the cells directly through H plus channels, bind to ligand-gated K plus channels, block exit of K plus, open ligand-gated channels for other positive ions, and allow them to diffuse into the cell. Bitter, stimulate through G protein mechanism. Umami amino acids like glutamate bind to receptors and depolarize through a G protein mechanism. 15.2 Taste. Neuronal pathways for taste, carried by three cranial nerves: chorda, tympani, part of facial nerve, carry sensations from anterior two-thirds of tongue, except from circumvallate papillae. Glossopharyngeal nerve carries taste sensations from posterior one-third tongue, the vallate papillae, and superior pharynx. Vagus nerve carries a few fibers for taste sensation from the tongue root and epiglottis. Pathways for the sense of taste. 1. Axons of cranial nerves extend from the taste buds to the tractus solitarius of the medulla oblongata. 2. Fibers from this nucleus extend to the thalamus, decussitating or desuscitating at the level of the midbrain not shown in figure. 3. Neurons from the thalamus project bilaterally to the taste areas of both hemispheres of the cerebrum. The taste areas are located in the insula, deep within the lateral fissure between the temporal and parietal lobes. Pathways for the sense of taste. Taste area of the insula. 1. Axons of the cranial nerve synapse in the tractus solitarius, nucleus of tractus solitarius, foramen magnum. Thalamus, taste area of the insula. 2. Axons extend to thalamus. 3. Neurons from thalamus synapse with taste area. Chorda, tympani, facial nerve. Trigeminal nerve, lingual branch. Glossopharyngeal nerve, vagus nerve. Figure 15.6. 15.3 Visual system. Visual system includes the eyes, accessory structures, and optic nerves. Tracts and pathways, accessory structures: eyebrows, shade, inhibit sweat. Eyelids, palpebrae with conjunctiva. Palpebral fissure, space between eyelids. Canthi, lateral and medial eyelids meet. Medial canthus has carbuncle with modified sweat and sebaceous glands. 5 layers of tissues, including a dense connective tissue tarsal plate that helps maintain shape of lid. 15.3 Visual system. Accessory structures of vision continued. Eyelashes, double, triple row of hairs. Ciliary glands, modified sweat glands empty into hair follicles. Tarsal glands at inner margins produce sebum. Conjunctiva, thin transparent mucous membrane covering eye and eyelids. Palpebral conjunctiva, inner surface eyelids. Bulbar conjunctiva. Bulbar conjunctiva, anterior surface of eye except over pupil. Structures of the eye. Superior palpebral eyelid. Superior palpebral eyelid. Pupil. Caruncle. Medial canthus. Eyebrow. Eyelashes. Iris. Sclera. Lateral canthus. Inferior palpebral eyelid. Figure 15.7a. Structures of the eye. Levator palpebrae superiors. Levator palpebrae superioris muscle, smooth muscle to tarsal plate. Superior rectus muscle, inferior rectus muscle, inferior oblique muscle. Eyebrow orbicularis oculi muscle, superior conjunctival fornix. Bulbar conjunctiva, palpebral conjunctiva. Tarsal meibomian gland. Tarsal plate, cornea, eyelash, palpebral fissure. Skin, areolar connective tissue. Orbicularis oculi muscle. Tarsal plate. Palpebral conjunctiva. Inferior conjunctival fornix. Lower eyelid inferior palpebra. Sagittal section. Figure 15.7b. 15.3 Visual system. Accessory structures of vision continued. Lacrimal apparatus consists of lacrimal gland and nasolacrimal duct. Tears are produced in the lacrimal gland and exit the gland through several lacrimal ducts onto the surface of the eyeball. Most of the fluid produced by the lacrimal glands evaporates from the surface. Excess drained by lacrimal canaliculi and into lacrimal sac. Tears flow from the lacrimal sac through the nasolacrimal duct into the nasal cavity. Lacrimal apparatus. Figure 15.8. 1. Lacrimal gland. lacrimal ducts. 2, 3, puncta, medial canthus, lacrimal canaliculi, lacrimal sact. 4, nasolacrimal duct. 15.3 Visual system. 6 extrinsic eye muscles are attached to each eye: superior, inferior, medial, lateral, rectus muscles, superior and inferior oblique muscles. Posterior, optic nerve, levator palpebrae superioris cut, lateral rectus, superior rectus, medial rectus, superior oblique, trochlea, figure 15.9. Extrinsic muscles of the eye. Superior, inferior. Levator palpebrae superioris cut, optic nerve, inferior rectus, B, lateral view. Trochlea, superior oblique, superior rectus, lateral rectus, inferior oblique, figure 15.9a. Extrinsic muscles of the eye. Superior rectus muscle, cut, eyeball. Medial rectus muscle. Lateral rectus muscle, optic nerve, optic chiasm. C, superior view. Figure 15.9c. 15.3 Visual system. The eyeball is a hollow, fluid-filled sphere. Wall is composed of 3 layers or tunics: fibrous tunic, sclera and cornea, vascular tunic, choroid, ciliary body, iris, nervous tunic, retina. Tunics and structures of the eyeball, sagittal section. Vitreous chamber filled with vitreous humor. Central retinal artery and vein, optic nerve, vitreous humor, retina, nervous tunic, choroid, vascular tunic, sclera, fibrous tunic. Conjunctiva, cornea, fibrous tunic, anterior chamber, posterior chamber, both filled with aqueous humor. Iris, vascular tunic, pupil, lens, suspensory ligaments, ciliary body, vascular tunic, figure 15.10. Figure 15.3 Visual system. Fibrous tunic consists of sclera. Fibrous tunic consists of sclera. Sclera, white outer layer, maintains shape protects internal structures, provides muscle attachment point, continuous with cornea, dense, collagenous, connective tissue with elastic fibers. Collagen fibers are large and opaque. 15.3 Visual system. Fibrous tunic consists of cornea, transparent window, continuous anteriorly with sclera, connective tissue matrix containing collagen, elastic fibers, and proteoglycans, layer of stratified squamous epithelium on the outer surface. Collagen fibers are small, thus transparent. Avascular, transparent, bends and refracts light. 15.3 Visual system. Vascular tunic contains most of the blood vessels of the eye, contains melanin. Iris, colored part of eye, determines pupil size. Sphincter pupillae, parasympathetic, CN3. Innervation, circular muscles, contraction decreases pupil size. Dilator pupillae, sympathetic innervation, radial muscles, contraction increases pupil size. 15.3 Visual system. Ciliary body, outer ciliary ring and inner ciliary processes. Ciliary muscles control lens shape, smooth muscle. Ciliary processes attached to the lens by suspensory ligaments produces aqueous humor that fills anterior chamber. Choroid, associated with sclera, thin and pigmented. Lens, cornea, iris, and ciliary body. Sclera, choroid, retina. Sclera, choroid, retina. Ciliary body, ciliary muscle, ciliary ring, ciliary processes. Vitreous chamber, suspensory ligaments, capsule of the lens, lens. Conjunctiva, sclerovenous sinus, canal of Schlemm, iris, posterior chamber, anterior chamber, cornea, outer epithelium, inner epithelium. Figure 15.11a. Lens, cornea, iris, and ciliary body. Lens, ciliary ring, ciliary processes, ciliary body, suspensory ligaments, sphincter pupillae, pupil, dilator pupillae. Figure 15.11b. 15.3 Visual system. The retina has two layers. Pigmented layer, outer layer, pigmented simple cuboidal epithelium. Pigment of this layer and choroid help to reduce light scattering. Neural layer, inner layer of rod and cone cells sensitive to light. Ophthalmoscopic view of the left retina. Optic disc, retinal vessels, macula, fovea centralis. Figure 15.12. Ophthalmoscopic view of the left retina. Lens focuses light on macula and fovea centralis. Macula, small yellow spot. Fovea centralis, area of greatest visual acuity, photoreceptor cells tightly packed. Optic disc, blind spot, area through which blood vessels enter eye, where nerve processes from neural layer meet and exit from eye. No photoreceptor cells here. 15.3 Visual system, chambers of the eye. Anterior chamber between cornea and iris. Posterior chamber between iris and lens. Aqueous humor fills anterior and posterior chambers. Helps maintain intraocular pressure and eyeball shape. Supplies nutrients to structures bathed by it. Contributes to refraction of light. Produced by ciliary process from blood, return to venous circulation through scleral venous sinus. 15.3 Visual system, glaucoma, abnormal increase in intraocular pressure. Vitreous chamber, posterior to lens, filled with jelly-like vitreous humor. Helps maintain intraocular pressure, holds lens and retina in place, refracts light. 15.3 Visual system. The lens is transparent and biconvex. Anterior surface lined with simple cuboidal epithelial cells. Posterior region contains long columnar epithelial cells called lens fibers. Lose nuclei and other organelles. Accumulate proteins called crystalline, covered by elastic transparent capsule, supported by suspensory ligaments. Functions of the eye. 1. As light passes through the pupil of the iris, it is focused on the retina by the cornea, lens, and humors. 2. The light striking the retina is converted into action potentials. 3. The optic nerve conveys these action potentials to the brain. Figure 15.13. 1. Light entering the eye is focused on the retina. 2. Action potentials initiated at the retina. 3. Action potentials conveyed to brain by the optic nerve. Visual system. Electromagnetic spectrum is the entire range of wavelengths or frequencies of electromagnetic radiation. Visible light, portion of electromagnetic spectrum detected by human eye, 380 to 750 nm, also called the visible spectrum. Electromagnetic spectrum, increasing energy, increasing wavelength, 0.001 nm, gamma rays 1 nm, X-rays 10 nm, UV light 1000 nm, infrared, visible light 380 nm, 430 nm, 500 nm, 560 nm, 600 nm, 650 nm, 750 nm. Infrared 0.01 cm, 1 cm, microwaves 1 m, radio waves 100 m, cm equal 10 minus 2 m, mm equals 10 minus 3 m, equals 10 minus net, and m equals 10 minus 9m. Figure 15.14. 15.3 Visual System. Refraction, the bending of light rays as they pass through materials of different densities. If the surface of a lens is concave, the light rays diverge as a result of refraction. If the surface of a lens is convex, the light rays converge as a result of refraction. Focal point where light rays converge and cross, focusing, causing light to converge. Lens changes shape, causing adjustment of focal point on the retina, produces an inverted image on the retina. 15.13 Visual System. Light rays converge as they move from the air to pass through the convex cornea. Greatest contrast and density here causes the most refraction. Additional convergence occurs as light passes through the aqueous humor, lens, and vitreous humor. Fine adjustments to focus light on the retina are made by changing the shape of the lens. 15.3 Visual System. Emetropia, normal resting condition of lens. Ciliary muscle is relaxed and lens is flat. Far point of vision, point at which lens does not have to thicken to focus, 20 feet or more from eye. Near point of vision, closer than 20 feet, point at which blurring occurs, changes occur in lens. Size of pupil and distance between pupils. Focus and accommodation. Fovea centralis, FP. Distant vision, ciliary muscles in the ciliary body are relaxed. Tension in suspensory ligaments is high. Lens flattened. A. Fovea centralis, FP. Near vision, ciliary muscles in the ciliary body contract, moving ciliary body toward lens. Tension in suspensory ligaments is low. Lens rounded. B. Figure 15.5. 15.3 Visual System. Events to focus for near point vision. Accommodation. Ciliary muscles contract due to parasympathetic input via cranial nerve. 3. Pull choroid toward lens, reducing tension on suspensory ligaments. Lens becomes more spherical, greater refraction of light. Pupil constriction varies depth of focus, allows the straightest light rays to enter the eye. Convergence, as objects move close to the eye, eyes are rotated medially. Reflex contraction of the medial rectus muscles. 15.3 Visual System. Visual acuity, ability to focus an image on the retina. Myopia, nearsightedness, cornea and lens too powerful or eyeball too long. Focal point in front of the retina, corrected by concave lens. Mild versions can be corrected by radial keratotomy and LASIK. 15.3 Visual System. Hyperopia, farsightedness, cornea and lens too weak or eyeball too short, focal point behind the retina, corrected by convex lens. Presbyopia, increase in near point of vision with age due to less flexible lens. Astigmatism, cornea, lens not uniformly curved so light rays do not focus at single focal point. Visual disorders and their correction. A. Myopia, nearsightedness, FP. B. Concave lens corrects myopia, FP. C. Hyperopia, farsightedness, FP. FP. D. Convex lens corrects hyperopia. Figure 15.16. 15.3 Visual system, structure and function of the retina. Neural layer has 3 layers of neurons: photoreceptor cells, bipolar cells, and ganglionic cells. Cell bodies form nuclear layers separated by plexiform layers where neurons of adjacent layers synapse with each other. Pigmented layer is a single layer of cells filled with melanin, along with choroid, enhances visual acuity by isolating individual photoreceptors, reducing light scattering. Retina. Choroid, pigmented layer, pigmented cell, photoreceptor layer, neural layer, outer plexiform layer, bipolar layer, inner plexiform layer, ganglionic layer, pigment cell, cone cell, rod cell, horizontal cell, bipolar cell, amacrine cell, interplexiform cell, ganglion cell, nerve fibers to optic nerve, direction of action, potential, propagation, direction of light, rod, cone. Figure 15.7. 15.3 Visual system, rods, bipolar photoreceptor cells, black and white vision, modified dendritic, light-sensitive part is cylindrical, contains about 700 double-layered membranous discs that contain rhodopsin, found over most of retina, but not in fovea, more sensitive to light than cones. Protein rhodopsin changes shape when struck by light and eventually separates into its 2 components, opsin and retinal. Retinal can be converted to vitamin A, from which it was originally derived. In absence of light, opsin and retinal recombine to form rhodopsin. 15.3 Visual system, rods continued. When not stimulated, rods are depolarized. Light causes them to hyperpolarize. Depolarization of rods causes depolarization of bipolar cells, causing depolarization of ganglion cells. Light and dark adaptation, adjustment of eyes to changes in light, happens because of changes in amount of available rhodopsin, pupil reflexes, and changes in level of photoreceptor function. Sensory receptor cells of the retina. outer segment, disc, inner segment, synaptic terminal, axons, cone, nuclei, disc, outer membrane, folding of outer membrane to form discs, outside of disc membrane, extracellular plug, opsin, retinal, inside of disc membrane, G protein, transducin, CGMP, phosphodiesterase, opsin, retinal, rhodopsin, dark configuration, gated and A plus channel, CGMP, figure 15.18. Rhodopsin cycle, figure 15.9. 11 CIS retinal, opsin dark configuration, 1 cross section, transducin G protein inactive, CGMP, phosphodiesterase inactive, Na plus channel open, CGMP, light, 2 opsin light configuration, all trans retinal, cross section, CGMP, phosphodiesterase active, transducin G protein active, GMP, CGMP, 3 Na plus channel closed, CGMP, phosphodiesterase inactive, 4 all trans retinal detaches from opsin, energy ATP, 5 CGMP Na plus channel opens, 6 11 CIS retinal, 7 rhodopsin cycle, 1 in the resting dark state, 11 CIS retinal is tightly bound to the internal surface of opsin. The extracellular plug helps keep retinal from dissociating from the opsin subunit through its channel. Cyclic GMP is attached to the Na plus channel, keeping it open. (11) CIS retinal, 1 opsin dark configuration, cross section, Na plus channel open, light, CGMP, transducin G protein inactive, CGMP phosphodiesterase inactive, figure 15.9. Rhodopsin cycle, 2 opsin light configuration, all trans retinal, cross section, CGMP, phosphodiesterase active, Na plus channel closed, 3 CGMP, GMP, transducin G protein active, figure 15.19. Rhodopsin cycle, 1 as rod cells absorb light, retinal changes shape from 11 CIS retinal to all trans retinal. This change causes opsin to also change shape dark to light state, the changes in opsin activate a G protein called transducin, which activates a cyclic GMP phosphodiesterase. To the cGMP phosphodiesterase catalyzes the conversion of cGMP to GMP. This reaction causes cGMP to diffuse away from Na plus channels, resulting in closure of the Na plus channels and hyperpolarization of the cell. Rhodopsin cycle 1. In the all-trans conformation, retinal detaches from the opsin molecule. Transducin reassociates with opsin, leading to the deactivation of cGMP phosphodiesterase. 4. Energy ATP. All trans retinal detaches from opsin. cGMP phosphodiesterase inactive. Figure 15.19. Rhodopsin cycle 1. In the absence of cGMP phosphodiesterase activity, cGMP concentration increases. cGMP binds to the Na plus channels, causing them to open again. 2. In a process requiring ATP, all trans retinal is converted to 11-CIS retinal. 3. In the 11-CIS conformation, retinal can attach to opsin, returning it to its original dark conformation. 11-CIS retinal, opsin dark configuration. 1. Cross-sectional. Transducin G protein inactive. cGMP phosphodiesterase inactive. cGMP. 5. cGMP Na plus channel opens. 6. 11-CIS retinal. 7. Figure 15.9. Rod cell hyperpolarization. 1. When photoreceptor cells are not exposed to light and are in a resting, non-activated state, gated Na plus channels in their membranes are open and Na plus flows into the cell. 2. This influx of Na plus, referred to as the dark current, causes the photoreceptor cells to release the neurotransmitter glutamate from their presynaptic terminals. 3. Glutamate binds to receptors on the postsynaptic membranes of the bipolar cells of the retina, causing them to hyperpolarize. Thus, glutamate causes an inhibitory postsynaptic potential, IPSP, in the bipolar cells. The influx of Na plus is offset by the efflux of K plus through non-gated K plus channels. Equilibrium of Na plus and K plus in the cell is maintained by a sodium-potassium pump. Rod cell hyperpolarization, figure 15.20a. Rhodopsin dark configuration. 1. cGMP phosphodiesterase inactive. Transducin G protein, open gated Na plus channel dark configuration. Na plus cGMP. Rod cell unstimulated. 2. Glutamate is continuously released. 3. Bipolar cell inhibited. Ganglion cell. Light pulse. MV -25, -30, -35. Hyperpolarization 1, 2, 3. Time S. Figure 15.20a. Rod cell hyperpolarization. 1. Exposure to light stimulates the rod cell. Rhodopsin and attached transducin are also activated. Transducin activates cGMP phosphodiesterase, which catalyzes the conversion of cGMP to GMP, closing Na plus channels. 2. As less Na plus enters the cell, glutamate release from the presynaptic terminal decreases. 3. The hyperpolarization in the bipolar cells decreases and they depolarize sufficiently to release neurotransmitters. 4. Bipolar cells release neurotransmitters that stimulate ganglionic cells to generate action potentials. Rod cell hyperpolarization. 4. Rhodopsin light configuration. Transducin active, closed gated Na plus channel, light configuration, Na plus, cGMP, GMP, light pulse, MV minus 25, minus 30, minus 35, hyperpolarization 1, 2, 3 time S. Rod cell hyperpolarized. 5. Glutamate release decreases. 6. Bipolar cell no longer inhibited. 7. Neurotransmitters released. Ganglion cell. Figure 15.20b. 15.3 Visual system. Mechanisms involved in light and dark adaptation. Changes in amount of available rhodopsin. In bright light, excess rhodopsin is broken down so that not as much is available to initiate action potentials, and the eyes become adapted to bright light. In a dark room, more rhodopsin is produced, making the retina more light sensitive. Pupil reflexes. In dim light, the pupil enlarges to allow more light into the eye. In bright light, the pupil constricts to allow less light into the eye. 15.3 Visual system. Mechanisms involved in light and dark adaptation continued. Changes in level of photoreceptor function during light conditions. Rod function decreases and cone function increases, whereas the opposite happens during dark conditions. This occurs because rod cells are more sensitive to light than cone cells and because the rhodopsin in rods is depleted more rapidly than the visual pigment in cones. 15.3 Visual system. Cones are responsible for color vision and visual acuity. Bipolar receptor cells with a conical light-sensitive part that tapers from base to apex. Outer segments consist of double-layered discs that are more numerous and more closely stacked than in rods. Requires bright light to function. Visual pigment is iodopsin, which consists of retinol combined with an opsin protein. Functions in the same way as the rhodopsin cycle. 3 types that respond to blue, red, and green light. Overlap in response to light, thus interpretations of gradation of color possible, several millions. Wavelengths to which blue, green, and red pigments are sensitive. Figure 15.21 Relative absorption percent of maximum 0, 10, 20, 30, 40, 50, 60, 70, 80, 90, 100. Blue pigment, green pigment, red pigment ALA 180, red pigment SER 180. Wavelength nanometers 400, 450, 500, 550, 600, 650. 15.3 Visual system. Inner layers of the retina. Rods and cones synapse with bipolar cells that synapse with ganglion cells in all areas except the fovea. Except in fovea centralis, ganglion cell axons converge at optic disc, then exit via optic nerve 2. Then impulses travel to visual cortex. Fovea centralis, highest visual acuity. Rods exhibit convergence. One bipolar cell receives input from numerous rods. One ganglion cell receives input from several bipolar cells. Cones exhibit little or no convergence. 15.3 Visual system, receptive field, area from which a ganglion cell receives input. Those in fovea smaller than in other parts of retina. Two types of receptive fields on center ganglion cells generate more action potentials when light is directed onto the receptive field. Respond to intensity of light. On center ganglion cells generate more action potentials when light is directed onto the receptive field. Respond to intensity of light. Off-center ganglion cells, more action potentials when light is off or when light does not hit center of field. Respond to contrasts in light. Interneurons present in inner layers and modify signal before signal leaves retina. Enhance borders and contours, increasing intensity at borders. Horizontal amacrine and interplexiform cells. Visual pathways. 1. Each eye has an area from which it collects light, referred to as the visual field. Each field is divided into the temporal part and the nasal part. 2. After passing through the cornea and lens, light from each half of the visual field projects to the opposite side of the retina. Stimulating photoreceptors, action potentials are conducted along the optic nerve. 3. The optic nerve leaves the eye and exits the orbit through the optic foramen to enter the cranial cavity. Just inside the cranial cavity and just anterior to the pituitary gland, the optic nerves are connected to each other at the optic chiasm. At this point, axons from some ganglion cells cross over and project to the opposite side of the brain. Visual pathways. 1. Specifically, ganglion cell axons from the nasal retina, the medial portion of the retina, cross through the optic chiasm and project. And specifically, ganglion cell axons from the nasal retina, the medial portion of the retina, cross through the optic chiasm and project to the optic. Specifically, ganglion cell axons from the nasal retina, the medial portion of the retina, cross through the optic chiasm and project to the opposite side of the brain. Ganglion cell axons from the temporal retina, the lateral portion of the retina, pass through the optic nerves and project to the brain on the same side of the body without crossing. This results in both hemispheres receiving visual input from both eyes. 2. Beyond the optic chiasm, the route of the ganglionic axons is called the optic tract. Most of the optic tract axons terminate in the lateral geniculate nucleus of the thalamus. However, some axons do not terminate in the thalamus but separate from the optic tract to terminate in the superior colliculi, the center for visual reflexes. 3. Neurons of the lateral geniculate nucleus of the thalamus form the fibers of the optic radiations, which project to the visual cortex in the occipital lobe. Visual pathways. Nasal parts of visual fields. 1. Temporal part of left visual field. Temporal part of right visual field. 2. Lens, left eye, temporal retina, lateral part. Nasal retina, medial part. 3. Optic nerves. 4. Optic chiasm. 4. Optic tracts. Superior colliculi. 5. Thalamus optic radiations. Visual cortex in each hemisphere. 6. Occipital lobe. Anterior, posterior. Optic nerve, optic chiasm, optic tract, thalamus, optic radiations, visual cortex. Optic nerve, optic chiasm, optic tract, thalamus, optic radiations, visual cortex. Figure 15.23. 15.3 Visual system organization of the visual field. Projections of ganglion cells from the retina of each eye are related to the visual field for each eye. Can be seen by observing with one eye closed. Divided into temporal, lateral, and nasal medial parts. Images from the right visual field projects to the left side of the brain. Binocular vision. Visual fields partially overlap, yielding depth perception. Organization of the visual field. Left visual field, temporal part of left visual field, right temporal part of left visual field, nasal part of left visual field, lens, left eye, temporal retina lateral part, nasal retina medial part, optic nerve, optic chiasm, optic tracts, superior colliculi, lateral geniculate nuclei of thalamus, optic radiations, visual cortex, left monocular, binocular, right monocular. Figure 15.24. 15.4 Hearing and equilibrium. Divided into external, middle, and inner ear. External and middle hearing. Internal hearing and equilibrium. External ear. Auricle, pinna, elastic cartilage covered with skin. External auditory meatus, lined with hairs and ceremonious glands that produce cerumen. Tympanic membrane, eardrum at boundary between external and middle ear. 2 layers of epithelia with connective tissue between. Sound waves cause it to vibrate. External, middle, and inner ears. Auricle, external ear, middle ear, inner ear. Temporal bone, external acoustic meatus. Tympanic membrane, chorda tympani, semicircular canals, oval window. Vestibulocochlear nerve, cochlear nerve, vestibule, cochlea, round window, auditory tube, auditory oculus in the middle ear. Malleus, incus, steps. 15.4 Hearing and equilibrium. Middle ear, separated from the inner ear by the oval and round windows. 2 openings provide air passages from middle ear. Auditory pharyngotympanic tube, Eustachian tube opens into pharynx, equalizes pressure between outside air and middle ear. Passage to mastoid air cells and mastoid process. 3 auditory ossicles, malleus, incus, stapes, transmit vibrations from eardrum to oval window. Oval window, connection between middle and inner ear. Foot of the stapes rests here, held in place by annular ligament. 15.14 Hearing and equilibrium. Middle ear continued. Skeletal muscles insert onto auditory ossicles. Tensor tympani. Tensor tympani attached to the malleus. Stapedius attached to the stapes. Act to dampen excessively loud sounds through the sound attenuation reflex. Auditory ossicles and muscles of the middle ear. Incus, posterior ligament of incus, chorda tympani nerve, stapedius muscle. Anterior, posterior, medial view. Superior ligament of malleus, head of malleus. Anterior ligament of malleus. Tensor tympani muscle. Auditory tube, tympanic membrane. Handle of malleus. Stapes. Figure 15.27. 15.4 Hearing and equilibrium. Inner ear contains fluid-filled tunnels and chambers. Bony labyrinth, tunnels and chambers in temporal bone. Cochlea involved in hearing. Vestibule and semicircular canals involved in equilibrium. Membranous labyrinth, membranous tunnels and chambers suspended in the bony labyrinth. Fluids within inner ear. Endolymph and membranous labyrinth, high K+, low Na+. Paralymph, in spaces between membranous labyrinth and bony labyrinth, low K+, high Na+. Inner ear, bony and membranous labyrinths. Figure 15.28. Bony labyrinth, membranous labyrinth. Semicircular canals. Oval window, round window. Vestibule, cochlea. Spiral lamina. Basilar membrane. Paralymph. Spiral ligament. Endolymph. Membranous labyrinth. Paralymph. Endosteum, boundary of bony labyrinth. Bony labyrinth, bone. Paralymph, fibrous bands from paralymphatic cells. Endolymph, membranous labyrinth. Bone, bony labyrinth. Endosteum, boundary of bony labyrinth. (15.4) Hearing and equilibrium. The cochlea has 3 regions: scala vestibuli, scala tympani, and cochlear duct. Scala vestibuli extends from oval window to helicotrema at cochlear apex. Scala tympani extends from helicotrema to round window. scala vestibuli and scala tympani filled with perilymph. Wall of scala vestibuli is vestibular membrane. Wall of scala tympani is basilar membrane. 15.4 Hearing and equilibrium. Structure of the cochlea continued. Cochlear duct formed by membranous labyrinth, space between vestibular and basilar membranes filled with endolymph. Width of basilar membrane increases from 0.04 mm near oval window to 0.5 mm near helicotrema. Near oval window basilar membrane responds to high frequency vibrations. Near helicotrema responds to low frequency vibrations. 15.4 Hearing and equilibrium. The structure of the cochlea continued. Spiral organ of Corti, located in cochlear duct, contains sensory cells for hearing. Sensory cells called hair cells with thin projections at the apical ends. These are microvilli called stereocilia. Inner hair cells primarily responsible for hearing. Outer hair cells regulate tension of basilar membrane. Tips of inner hair cells embedded in tectorial membrane. Basilar region of hair cells covered by synaptic terminals of sensory neurons. Cell bodies of afferent neurons grouped into cochlear ganglion. Afferent fibers from the cochlear nerve. Cochlea. Semicircular canals, vestibule, oval window, cochlea, round window, helicotrema, cochlear nerve, bone boundary of bony labyrinth, membranous labyrinth, spiral lamina bone, cochlear ganglion, paralymphatic cells, endosteum, scala tympani filled with perilymph, basilar membrane, spiral ligament, cochlear duct filled with endolymph, tectorial membrane, vestibular membrane, scala vestibuli filled with perilymph. Figure 15.29 A, B. Cochlea, vestibular membrane, tectorial membrane, stereocilia, cochlear nerve, spiral lamina bone, hair cell, nerve endings of cochlear nerve, supporting cells, outer hair cells, inner hair cell, spiral organ, spiral ligament, basilar membrane, cochlear duct. Scanning electron micrograph of cochlear hair cell, stereocilia. A, B, tip links, stereocilia. C, stereocilia, tip links. D. Wow. Figure 15.30 A, D. Figure 15.30 A through D. Cochlear hair cell stereocilia. Tip link gating spring attaches tip of each stereocilium in a hair bundle to the side of the next longer stereocilium. As stereocilia bend, they open K plus gates, mechanically gated ion channels. Gating spring gate K plus channel K plus. Relaxed tip link, stretched tip link K plus. (15.4) Hearing and equilibrium. Auditory function. Vibrations of matter, air, water, solids produce sound. When a person speaks, vocal cords vibrate, air passing out of lungs vibrate, vibrations propagated through air as sound waves. Volume, loudness, function of wave, amplitude, height measured in decibels. The greater the amplitude, the louder the sound. Pitch, function of wave frequency. The higher the frequency, the higher the pitch. Timbre, resonance quality or overtones of sound. Amplitude, volume, one cycle of tuning fork. Tuning fork. Compressed air, less compressed air, compressed air. Amplitude, volume. Lower amplitude, lower volume. Time, amplitude, volume. Lower frequency, lower pitch. Higher frequency, higher pitch. Figure 15.3. 15.4 Hearing and equilibrium. The process of hearing. External ear collects sound waves, conducts through external auditory canal. Tympanic membrane vibrates. Ocellus vibrates. Vibrations transferred to oval window. Vibration of perilymph causes vestibular membrane to vibrate, which causes vibrations in endolymph. Basilar membrane displaced, detected by hair cells. Vibrations in scala tympani, dissipated by movement of round window. Effect of sound waves on cochlear structures. Oval window, steps, incus, Milius, tympanic membrane, external acoustic meatus, round window, auditory tube, oval window, stapes, incus, Milius, tympanic membrane, external acoustic meatus, round window, auditory tube. 1. External acoustic meatus. 2. 3. 4. 5. 6. 7. Cochlear nerve. Space between bony labyrinth and membranous labyrinth contains perilymph. Scala vestibuli, scala tympani. Space between bony labyrinth and membranous labyrinth contains perilymph. Cochlear duct contains endolymph. Membranous labyrinth. Vestibular membrane, basilar membrane. Tectorial membrane, spiral organ. Figure 15.32. Effect of sound waves on points along the basilar membrane. Different frequency sounds cause maximum vibrations of different regions of basilar membrane. High pitch near base, low pitch near apex. Highest pitched sounds, lowest pitched sounds. $20,000 Hz. $7,000 Hz. $5,000 Hz. 4,000 Hz, 3,000 Hz, 1,500 Hz, 600 Hz, 800 Hz, 1,000 Hz, 200 Hz. Bass, 7,000 Hz, high frequency sounds, 0.04 mm, 1,000 Hz. 200 Hz, low frequency sounds, apex, relative width of basilar membrane, 0.5 mm, basilar membrane. Figure 15.33. 15.4 Hearing and equilibrium. Creation of an action potential during the process of hearing. 1. Apical portion of hair cell surrounded by endolymph, while the basal portion is surrounded by perilymph. 2. Endocochlear potential, charge between the endolymph and perilymph. 3. When the potassium channels open when a hair cell bends, potassium flows into the hair cell, attracted to the negative charge inside the hair cell. 4. This inward movement of potassium causes the slight depolarization of the hair cell. 5. Voltage-gated calcium channels open to allow calcium to flow into the cell, causing further depolarization. 15.4 Hearing and equilibrium. 1. Neurotransmitter glutamate is released, which increases the action potential frequency at the basal region of the hair cell. 2. This also opens voltage-gated potassium channels in the basal region, allowing potassium to leave the hair cell, causing repolarization. 3. Neurotransmitter release from the inner hair cells induces action potentials in the sensory cochlear neurons that synapse with the hair cells. 15.4 Hearing and equilibrium. Fine-tuning tension on basilar membrane. More than 90% of afferent axons of the cochlear ganglion synapse with inner hair cells, 10 to 30 hair cells. A few small diameter afferent axons synapse with rows of outer hair cells. Outer hair cells receive efferent inputs, causing them to shorten, tuning hair cells to specific frequencies. Actin filaments in hair cells attach to K+ gated channels and can move them along the cell membrane and tighten or loosen the spring. Hair cells tune to very specific frequencies. 15.4 Hearing and equilibrium. Localization of pitch along the cochlea. Afferent cochlear nerve fibers send action potentials to superior olivary nucleus and medulla oblongata. These are compared to one another, and strongest is taken as standard. Afferent action potentials inhibit other action potentials. Action potentials from maximum vibration go to cortex and are perceived. 15.4 Hearing and equilibrium. Neuronal pathways for hearing. The neurons from the cochlear ganglion synapse with CNS neurons in the dorsal or ventral cochlear nucleus. These neurons either synapse in or pass through the superior olivary nucleus. Neurons terminating in the nucleus may synapse with different Neurons terminating in the nucleus may synapse with efferent neurons returning to the neurons terminating in the nucleus may synapse with efferent neurons returning to the cochlea to modulate pitch perception as described earlier. Nerve fibers from the superior olivary nucleus also project to the trigeminal nerve nucleus, which controls the tensor tympani and the facial nerve nucleus, which controls the stapedius muscle. 15.4 Hearing and equilibrium. Neuronal pathways for hearing continued. Ascending neurons from the superior olivary nucleus travel in the lateral lemniscus. All ascending fibers synapse in the inferior cochlear. All ascending fibers synapse in the inferior colliculi, and neurons from their project to the medial geniculate nucleus of the thalamus, where they synapse with neurons that project to the cortex. These neurons terminate in the auditory cortex. Neurons from the inferior colliculus also project to the superior colliculus, where reflexes that turn the head and eyes in response to loud sounds are initiated. Central nervous system pathways for hearing. Auditory cortex, thalamus. 1. Cochlear nucleus. Nerve stapedius. Vestibular cochlear nerve. Cochlear ganglion, nerve to tensor tympani. Superior olivary nucleus. 2. Inferior colliculus. 3. Auditory cortex. 4. Thalamus. Auditory cortex. Medial geniculate nucleus. Frontal section, figure 15.34. 15.4 Hearing and equilibrium. Organs of equilibrium divided into two parts, static and dynamic labyrinths. Static labyrinth consists of utricle and saccule of the vestibule. Evaluate static equilibrium. Position of head relative to gravity. Also detects linear acceleration and deceleration, as in a car that is increasing or decreasing in speed. Dynamic labyrinth, crista ampullaris of the semicircular canals, evaluates dynamic equilibrium, movements of the head. 15.4 Hearing and equilibrium. Static labyrinth, utricular macula oriented parallel to base of skull. Saccular macula oriented perpendicular to base of skull. Each consists of supporting cells and hair cells with numerous stereocilia, microvilli, and one cilium, kinocilium, embedded in gelatinous mass weighted by otoliths. Gelatinous mass moves in response to gravity, bending hair cells and initiating action potentials. Otoliths stimulate hair cells with varying frequencies. Pattern of stimulation translated by brain into specific information about head position or acceleration. Structure of the utricular and saccular maculae. Figure 15.35. A. Vestibule, utricle saccule, saccular macula, utricular macula. B, part of macula, hair cell supporting cells. C, nerve fibers of vestibular nerve, canocleum, stereocilia, microvilli. Otolithic membrane. D, figure 15.35. Function of the vestibule in static equilibrium. A, endolymph in utricle, otolithic membrane, hair cell, supporting cell, vestibular nerve fibers, macula. B, force of gravity. Figure 15.36. 15.4, hearing and equilibrium. A dynamic labyrinth. 3 semicircular canals filled with endolymph. 1 in the transverse plane, 1 in the coronal plane, and 1 in the sagittal plane. Base of each expanded into ampulla with sensory epithelium, crista ampullaris. Cupula suspended over crista hair cells, acts as a float displaced by fluid movements within semicircular canals. Displacement of the capula is most intense when the rate of head movement changes. Thus, the system detects changes in the rate of movement rather than movement alone. Semicircular canals, figure 15.37. Semicircular canals, cupula, vestibular nerve, ampullae. A, B, cupula, stereocilia, crista ampullaris, nerve fibers to vestibular nerve, hair cell. C, function of the semicircular canals, figure 15.38. Endolymph in semicircular canal causes movement of the cupula, hair cell, crista ampullaris. Movement of semicircular canal with body movement. (15.4) Hearing and equilibrium. Neuronal pathways for equilibrium. Neurons synapsing on their hair cells of the maculae and cristae ampullaris converge into the vestibular ganglion, where their cell bodies are located. Sensory fibers from these neurons join sensory fibers from the cochlear ganglion to form the vestibulocochlear nerve, terminate in vestibular nucleus of medulla. Axons run from this nucleus to numerous areas of CNS, such as the spinal cord, cerebellum, cerebral cortex. 15.4 Hearing and equilibrium. Neuronal pathways for equilibrium continued. axons from the vestibular nuclei synapse within motor nuclei of the oculomotor, trochlear, and abducens cranial nerves, which control reflexive movements of the extrinsic eye muscles. Vestibular neurons also send axons to the posterior ventral nucleus of the thalamus. Thalamic neurons project the vestibular areas of the cerebral cortex, allowing for conscious awareness of equilibrium. Central nervous system pathways for equilibrium. One, vestibular nucleus, spino-vestibular tract, vestibulospinal tract, frontal section. 2, vestibular ganglion, vestibular nerve, vestibulocochlear nerve, cerebellum, posterior ventral nucleus, thalamus, abducens motor nucleus, trochlear motor nucleus, oculomotor nucleus. 3, 4, 5, vestibular area, figure 15.39. Effects of aging on the special senses. Slight loss in ability to detect odors, decreased sense of taste, lenses of eyes lose flexibility, presbyopia, development of cataracts, macular degeneration, glaucoma, diabetic retinopathy, decline in visual acuity and color perception, presbyacusis, age-related hearing loss, hair cells in the cochlea, utricle, saccal, and ampulla decrease, more falls due to instability and vertigo. The time now is 8:30 p.m. The date today is Wednesday, August 19th, 2026. Assalamu alaikum wa rahmatullahi wa barakatuh.